everyone, I'm Riyang Li, a freshman from the Kunshan University. Have you ever heard of the DKU Bio110? I'm taking this course at the moment and I love it very much. My professor is Johann Li. Professor Li is a molecular geneticist with a PhD from Dartmouth College, and he has served as an experienced faculty member at Amherst College before joining at Kunshan University. The course itself focuses mainly on integrated science and is designed around the connections between the disciplines, allowing us to experience both the depth and the breadth of the scientific theory and practice. This session's Bio110 employs 5 themes that describe properties of life. The first one is the organization, about structure and function. The second is cycling of energy and matter. The third is information, about genetic variation. The fourth is homeostasis, about interactions. And the last is evolution. These themes are unified under the organizational principles of the scientific method, formulating hypotheses and testing hypotheses with experiments. And today, I'll mainly focus on one of the lab experiments I've recently done, which is bacterial transformation. Above all, what is transformation? Transformation is a fascinating process in biology where bacteria acquire new DNA and gain new characteristics. In this lab, we will transform E. coli bacteria using a plasmid called PUC19. This plasmid contains 2 important genes, the ampicillin resistance gene AMPR and the LACZ gene, which encodes beta-galactosidase. When transformed, these bacteria will not only survive on ampicillin, but also turn blue in the presence of IPTG and X-scale. Let's see how this works in practice. So, welcome to today's lab. Now we'll be diving into the 5th lab of Bio110, bacterial transformation. This experiment will help us understand the key concepts like plasmids, competent cells, cell transformation, and transformants. We also perform a hands-on transformation using PUC19 plasmid and DH5-alpha competent cells, and observe the growth of bacteria on antibiotic-containing agar plates. Let's get started and explore each step of this exciting experiment. Before we begin, let's take a moment to familiarize ourselves with the materials. At each workstation, you will find 2 types of agar plates, LB IPTG X-scale plates with and without ampicillin. These will help us distinguish between transformed and non-transformed bacteria. You also need a tube of LB liquid media, several microcentrifuge tubes, and a set of micropipettes with tips P20, P200, and P1000. These are essential for precise volume transfers. Additionally, you have a tube of sterile glass beads for spreading bacteria on the plates, and an ice bucket with crushed ice to keep your component cells cold, and a marker for labeling. Make sure to check your workstation for all these supplies before starting. Clean your bench thoroughly with 75% ethanol to ensure a sterile environment. Label your tubes and plates clearly, 1 tube for the plasmid plus and 1 without minus. For the plates, label them according to the conditions. LB AMP plus PUC19, LB AMP minus PUC19, LB plus PUC19 and LB minus PUC19. This carefully labeling will help you track your results accurately and avoid confusion during the experiment. Remember, proper preparations is a key to a successful transformation. First, label 2 tubes, 1 for the plasmid and 1 without. Keep them on ice. Next, add 50 microliters of competent cells to each tube. To the plasmid tube, add 10 microliters of PUC19 plasmid solution. Incubate both tubes on ice for 20 minutes. After that, heat shock the tubes in a 42 degrees Celsius water bath for exactly 45 seconds and then return them to ice for 2 more minutes. This step helps the cells take up the plasmid efficiently. Next, add 500 microliters of LB media to each tube. Mix gently and incubate at 37 degrees Celsius for 20 minutes. Finally, spin down the cells, resuspend them in the remaining liquid, and plate 100 microliters of each solution onto the corresponding agar plates. Use stereo glass beads to spread the cells evenly. After plating, stack your plates, tap them together, and incubate them upside down at 37 degrees Celsius overnight. Next day, observe the plates and record your results. You should see blue colonies on the plates with POC19 and ampicillin, indicating successful transformation. The control plates without ampicillin or plasmid will help you understand the baseline growth of the bacteria. When doing the experiment, my hypothesis was that DH5 alpha competent cells would successfully take up the POC19 plasmid, leading to the formation of ampicillin-resistant colonies that express beta-glycosidase, turning blue on LB, AMP, IPTG axial plates. The expected results were blue colonies on selective plates and no colonies on the negative control plates. The actual data largely supported our hypothesis, with blue colonies observed on selective plates indicating the successful transformation. The absence of colonies on negative control plates validated the specificity of our method. However, the unexpected lack of blue color on colonies in the LB IPTG axial plates without ampicillin suggested the potential issues with IPTG induction or axial substrate availability. Further experiment could vary IPTG concentrations or incubation times to optimize beta-glycosidase expression. Additionally, testing different groups of axial could help determine if substrate quality affected the results. These modifications could enhance our understanding of bacterial transformation efficiency and its applications in genetic research and biotechnology. Based on these results, further studies can also explore the long-term stability of transformed cells and their implications for antibiotic resistance. That's all for the experiment section. Before the end of the video, I would like to take a moment to express my gratitude. First and foremost, I want to thank my family for their unwavering support and encouragement throughout my studies at Kunshan University. I'm also incredibly grateful to DKU for providing such a rich and inspiring learning environment. The resources and opportunities here have truly enhanced my educational experience. A special thank you goes to Professor Johan Lee from the Bio110 course. Your guidance and passion for biology have made this subject even more fascinating for me. You have been patient and always willing to help, and I appreciate it more than words can say. That's all for this video, and thank you for watching.